Assalamu alaikum, dear students. Let's discuss the third example of enzyme in our protein therapeutic section. And the enzyme I'm talking about is phenylalanine ammoniase. This enzyme is of therapeutic importance for a phenotype known as phenylketonuria. Phenylketonuria is an autosomal recessive genetic disorder which arises because of mutant phenylalanine hydrolase gene. Um, the frequency of this disorder is 1 in 12,000 in the United States. And to date, 500 different mutations are reported in the said gene, indicating it has allelic heterogeneity. So what happens when our pH enzyme, which is phenylalanine hydrolase, if it in the natural setup, it oxidizes phenylalanine to tyrosine, but when it is deficient or mutant, then it won't be able to oxidize phenylalanine to tyrosine. As a result, the normal cognitive development of an individual is impaired, and that results in the mental retardation, which ultimately is due to the build-up of phenylalanine in the system. Kindly keep in mind that phenylalanine hydrolase is a multi-enzyme complex which is not very stable, and it requires the cofactor tetrahydrobiopterin for its activity. I have shown it in the slide as well. So this slide tells us briefly about what phenylketonuria is and how it is transmitted in autosomal recessive manner and what it actually does, like conversion of phenylalanine to tyrosine, and that tyrosine then ultimately converts into dopamine. So if the tyrosine is not present, it is not being formed from phenylalanine, then it leads to many other brain related impairments. Which foods are rich in phenylalanine? These are meat, eggs, and even the artificial sweetener aspartame is rich in phenylalanine. And when the level of phenylalanine is high, then the people may have difficulties with attention and cognitive functions. For example, you can call it as brain fog. That they said that we don't feel active enough to respond to different situations. For others, the high levels of phenylalanine may result in anxiety or depression. One can also identify phenylketonuria with the help of urine tests. What happens that the urine may appear green or brown when ferric chloride solution is added to the patient's urine, and this color change occurs because the excess phenylalanine in the urine reacts with the ferric chloride. But when there is a normal person, then addition of ferric chloride won't turn it into um, dark green or brown color. Additionally, their skin is also musty, and there is a strange musty mousy odor which comes from the urine of such patients. Now the problem which arose in front of the scientist in the past decades was that depending upon the source of the pH, uh, the therapies like enzyme replacement therapy, it might be immunogenic. It might cause immunogenic responses in the patient that further leads to severe problems in the phenylketonuria patients. So there were multiple solutions to this problem. First one was, which you have already studied, and I think you can propose it too, and that is pegylation of the pH enzyme. And the second solution was phenylalanine ammoniase. Now, what this PAL or phenylalanine ammoniase enzyme do? It converts phenylalanine to ammonia and tranexamic acid. Now, this enzyme, our PAL enzyme, is a stable enzyme as compared to the pH enzyme. The pH enzyme is not only unstable, but it also requires a cofactor, which I have told you in the previous slide. But when we talk about the enzyme PAL, it is a stable enzyme which does not require a cofactor. And ultimately, the cinnamic acid which is being produced converts into hippuric acid, and that hippuric acid can be excreted in the urine. We can we can express the PAL gene uh, in the biological system. We can do its biotechnological expression. For that, what the scientists did in the past, they extracted the PAL gene from the yeast strain, and the yeast strain they used was Rhodosporidium tauroloides, and they extracted gene from or amplified gene from this yeast species, and then they cloned it and overexpressed in the host cell, which was the prokaryotic host E. coli. So after that, they did preclinical studies with the PAL defective mice, which has accumulated phenylalanine. When these mice were treated with the PAL, the recombinant PAL, which was being produced in the E. coli, the plasma phenyl levels were lowered. And what they did, they injected this PAL intravenously, or they also tried it giving in the encapsulated form, which can be given orally with the feed. So, at least in the mice, the recombinant PAL was identified as an effective substitute for pH, and orally delivered enzyme is sufficiently stable to survive the mouse gastrointestinal tract and remain functional. Although I must say, although this report is preliminary, but a combination of oral enzyme therapy and a less stringent low phenyl diet may ultimately will help or serve to improve the quality of life of phenylketonuria patients. And when it comes to the second one, pegylated PAL, it is in the phase 2 clinical trials as per the book which we are consulting or referring to, which is Biotechnology by Glegg, and its uh, latest edition, which came in 2017.